Biosimilars Opportunities and Challenges by MentorAndServe.com.
Hi friends, let us see what these biosimilars are and what is their scope, importance, and future. Let us see what they are. Therapeutic or preventive medicine derived from living cells, using recombinant DNA technology. Conventional pharmaceuticals are generally small molecules. Biopharmaceuticals are typically proteins, peptide, nucleic acids, or inactivated viruses, bacteria. These are the families of biopharmaceuticals. And these are the differences between conventional drugs and biopharmaceuticals. IMS have published this data in 2009 about top biopharmaceuticals in industry. The erythropoietin products have largest share of market among all top 10 players in biotech field. These are not all but prominent players in the field: Amgen, Roche, Genentech, Johnson & Johnson, Novo Nordisk, Lilly, Sanofi Aventis, Abbott, Serrano Merck, Schering Lab, Wyeth. Biosimilars. What are they? Actually, they are biosimilars, biogenerics, follow-on biologics, follow-on proteins, follow-on innovators products developed by parties other than original developers using either identical or different manufacturing processes. The products must be bioequivalent or comparable to the original innovative products in terms of preclinical, PARC, PG, as well as safety and efficacy. The global demand, as we can see here, increasing with time. How is a biosimilar product developed? Let us see it in detail. The actual process of development. In upstream, it involves cell line development, fermentation, while in downstream, purification, fill and finish. How is a biosimilar product developed? Comprehensive analytical characterization and comparability study, clinical study, preclinical, clinical, submission. 1. Cell line development. The fundamental. Analyze the DNA sequence of the targeted protein based on the published literature information. Synthesize the cDNA. Construct the vector. 1. Cell line development. The fundamental. Transfer the vector into the cell. Positive cell line screening and selection. High-yield cell line selection. Cell bank characterization and storage. Master cell bank and working cell bank. 2. The fermentation process development. Bioreactor selections. Culture medium optimization. High-yield culture conditions. Process scale-up, difficulty. Extensive comparability study need to be done. 3. Protein purification. It involves cell broking, removal of cells, capture of proteins, removal of virus, 3. Protein purification. Primary purification. Secondary purification. Lyophilization of purified proteins. Characterization. 4. Fill finish. Formulation development. Freezing, thawing impact. Manufacturing process development. Mixing and holding time. Filter compatibility. 4. Fill finish. Microbial retention. Lyocycle development. Package selection. Device development. Stability testing. 5. Extensive comparability study. 1. With innovator products. 2. When cell line changes. 3. When culture medium changes. 4. When culture condition changes. 5. Extensive comparability study. When master and working cell bank generation changes. When fermentation process changes. When scaled up. When facility changes. Using stressed samples. 6. Clinical trials, lengthy and expensive. Preclinical study. Toxicology in animals. Phase I study. PARC, PD in healthy subjects. 6. Clinical trials, lengthy and expensive. Phase III study. Safety, efficacy. All indications. In patients. Immunogenicity study. Post-market monitoring. 7. Submissions. No clear regulatory pathway in U.S. Product specific in EU. FDA treated as BLA. Biological license agreement. 7. Submissions. Pre-IND meetings and scientific advisory. Meeting are needed. Expensive submission fee. Challenges continued. No interchangeability, brand marketing required. Injectable manufacturing capability. Microbial and mammalian cell fermentation facilities in large scale. Overall, high barriers to entry, but high margin. Public demanding for affordable biological drugs. Challenges continued. High cost, longer term. Technology derived. Complicated analytical techniques. Innovative like clinical studies. Defective brand patent protection for over 20 years. Enter the market at risk. No paragraph 4. Summary. Biosimilars are the trend. Have drawn extensive attentions from public, regulatory bodies, governments and certainly pharmaceutical companies. Challenging, cost, but worth to develop. Has created huge opportunities for the organizations and CRO who are engaged in the analysis and characterizations of biological molecules and cell culture. Thanks for watching. Visit us at mentoranserve.com. Biosimilar